My name is Dr. Richard Stone, and I'm the director of the Adult Leukemia Program at the Dana-Farber Cancer Institute in Boston, Massachusetts. I'm also the chair of the Leukemia Committee of the Alliance Cooperative Group in the United States. So we know that AML in general is an adverse prognosis disease, but there's a subgroup with an even worse prognosis, and those would be the approximately 35% of patients with AML that have a FLT3 mutation. There are different subtypes of mutations. One of those, an internal tandem duplication mutation, is particularly bad. Anyway, that's a bad outcome, unmet need. And we had a drug, namely midostaurin, that targets the enzyme that's abnormal in this type of patient population. The drug is not the cleanest drug in the world, which may be good. It also hits other enzymes which might be relevant in the pathophysiology of AML. We knew that as a single agent in relapse refractory patients with AML, it had biological activity but not major clinical activity. But it worked a little bit. We also knew from preclinical and early clinical studies that combining midostaurin with standard chemotherapy seemed to be better than chemotherapy alone, although we needed to do a randomized trial to prove that fact. And that was the genesis of CALGB 10603, or the RATIFY trial that we're talking about today. The first point is we had to screen almost 3,300 patients to see who had a FLT3 mutation and who didn't, because only those with a FLT3 mutation were eligible for the trial. So that was part one. It was a targeted, personalized medicine type of deal, and to get the answer rapidly in this very sick group of patients so we wouldn't delay their therapy, we were able to use 9 academic labs around the world to tell us who had a mutation and who didn't. So that's part one. And so it was simply chemotherapy alone, like we normally give, with placebo or with midostaurin. And the outcome was that there was a survival advantage for people who were randomized to midostaurin, 23% reduction in the risk of dying if you were randomized to the midostaurin arm. There was also improvement in event-free survival, an improvement in overall survival if you censored for transplant, because transplant was very common, and an improvement in disease-free survival as well. We compared the toxicities in the two arms, of course. There were no increased deaths in the midostaurin arm on treatment. The only increased toxicity we saw for midostaurin randomized patients was an increased incidence of serious rashes, but that wasn't that common, about 12% versus 8% in the placebo group. I believe that AML patients and their treating doctors will review this as an advance, and I know the drug company that owns midostaurin, namely Novartis, is going to try to get this approved by the FDA so it can be used routinely. In the meantime, I think they're readying a compassionate use program so that FLT3 positive AML patients will have access to it perhaps even before it's officially approved.